Hello, my dear students. Today we are discussing the important questions that is from the chapter Principles of Inheritance and Variation. So, here already we have discussed the multiple choice question in the first session. So, we are discussing the 2 marks and 5 mark question in this session. Okay. So, this is one of the important chapter as I said. Totally 13 marks question will be asked from this chapter. Okay, one of the very important chapter for your board exam. So here in today's session, we are going to discuss the 2 mark question as well as 5 mark question. So 2 5 mark questions will be asked from this chapter. So we are concentrating on both session. Okay. Yes, let us start with the first question, first 2 mark question. Derive, derive the phenotypic and genotypic ratio of a cross between AB blood group parents. Here we have to derive the phenotypic and genotypic ratio of the AB blood group parents. Okay. Cross between the AB blood group parents. Here AB blood group means they have the IA and IB alleles, right? IA and IB genetic type, IA and IB. Okay, so this is IA and IB. When they are crossed, when these parents are crossed, the progenies, that is IA, then IA, IB, IA, IB, then this one IA, IB, IA, IB, then IB. Last one is the IB. Okay. Here they asked phenotypic ratio. Phenotypic ratio, that is they are having the A, AB and B blood group in the ratio of 1:2:1. So this is the phenotypic ratio of the blood group. A, AB and B blood group, they are in 1:2:1 ratio. Then what is the genotypic ratio of these blood groups? That is genotypic ratio. Here A blood group IA blood group 1, then IAIB means B blood group, AB blood group 2 and IB, right? So that is also 1. Totally 4 blood groups, that is also 1:2:1. The ratio of the A, AB and B blood group phenotypic ratio is 1:2:1 and genotypic ratio also 1:2:1. Okay, that is the correct answer. Here you have to derive the phenotypic and genotypic ratio of the these two parents. Okay. Next question. What is haplodiploid sex determination mechanism? Mention animal which exhibits this. Okay. So this type of the haplodiploid type of the sex determination done in honeybee. Okay. So this haplodiploid type of the sex determination done in honeybee. Here, how it done haplodiploid type of the sex determination? Here, the sex determination in honeybee is based on the number of sets of chromosomes in individual uh, sets of chromosomes and individual receives. Okay, an offspring formed from the union of the sperm and an egg develops as a female. When the sperm and egg, so that is the sperm and egg, when they fuse, they will develop, always the zygote will develop into female and unfertilized male sperm. So the unfertilized egg, the unfertilized egg that develops into a male. Okay, so here males, they are grown by the parthenogenesis. They developed by the parthenogenesis. This means that the males have half number of chromosomes than that of the female, because females are formed by the fusion of the male and female gametes, right? But the males are formed by the only unfertilized egg. So they are having the only half set of the chromosomes. Here the females are diploid having the 32 chromosomes, and males are haploid, they are having the 16 chromosome. Okay, this is called haplodiploid type of the sex determination. So this is the special type of the sex determination. Here males produce sperms by the mitosis. So these males are haploid, they produce the sperms by the mitosis process. They are having the 16 number of chromosomes. So they do not have father. These males, they do not have father, and thus cannot have sons. They will not have sons but have grandfather and can have the grandsons. Okay, so like that, the haplodiploid type of the sex determination done in honeybee. Very important question. Write a short note on sex determination method in birds. So this is also one of the important question. Write a short note on sex determination method in birds. Here, in many birds, female has pair of dissimilar chromosomes, that is ZW chromosomes, and males, they are having the ZZ similar chromosomes. So this type of the sex determination is called, this type is called as a female heterogamety. Means females are able to produce the two types of the gametes, so which may contain Z chromosome or W chromosome, but the male gametes, they contain always Z chromosome. Okay, so this type of the sex determination done in the birds, female heterogamety is present. When the Z gamete of the female fuses with the Z gamete of the male, it develop into a male. When the W gamete of the female fuses with the Z gamete of the male, then it will develop into a female. Okay, so this is where the 50% of the chances of the fusion with the Z, and 50% chances of fusion with the Z gamete with the W chromosome. Okay, so like this, the female heterogamety is observed. So you should explain this. Okay, ZW, so gametes ಅಲ್ಲಿ ಯಾವ್ದಿರುತ್ತೆ, Z W ಮತ್ತೆ Z ಫ್ಯೂಷನ್ ಆದ್ರೆ ಏನಾಗುತ್ತೆ ಅದನ್ನ ನೀವು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಬೇಕು. Next question. Differentiate between linkage and recombination. Here, first, linkage. The phenomenon where genes located close to each other on the same chromosome tend to be inherited together because they are physically connected. That is called as a linkage. So, dear students, while writing the difference, make the two columns and write in one column linkage and recombination. Write the points which is related to linkage and the points which are related to the recombination in each column. Okay, so like that, while writing the difference, if you write like that, it carries the good marks. So but here I'm not giving the points like that. I'm giving the total what is linkage and what is recombination, but you can make the short note like that. Okay, then what is recombination here? Recombination is the process that creates a new combinations of alleles by exchanging the genetic materials between the homologous chromosomes during the meiosis. They exchange the genetic material between the homologous chromosomes. But here there is no exchange of the homologous genetic material between the homologous chromosomes. Okay, here, based on physical proximity of genes on a chromosome, genes that are tightly linked are less likely to be separated during the meiosis. Here, it is based on the crossing over where the homologous chromosomes exchange their segments during the prophase 1 of the meiosis. And here, it preserves the existing combination of alleles, maintaining the parental gene combination in offspring. So, parentally ಯಾವ gene combination ಇರುತ್ತೆ ಅಲ್ವಾ ಅದನ್ನ offspring ಅಲ್ಲೂ ಕೂಡ maintain ಮಾಡಲಿಕ್ಕೆ. So, this linkage is helping. But recombination ನಲ್ಲಿ ಏನಾಗುತ್ತೆ recombination generates the genetic diversity by creating the new allele combination. ಅಲ್ಲೇನಾಗುತ್ತೆ recombination ಆಗೋದ್ರಿಂದ new allele combination and new characters will develop and it promotes the evolution. Okay, so that is the main difference between the linkage and recombination. Next question. What is sickle cell anemia and why RBCs become sickle shaped in this condition? So read the question properly. This is the 2-mark question. What is sickle cell anemia and why RBCs become sickle shaped in this condition? Here, sickle cell anemia is an autosome linked recessive trait that can be transmitted from parents to the offspring when both partners are carrier for the gene. So they are called as heterozygous. They are carriers. They are called as a heterozygous. Then when the sickle shaped RBCs form, the mutant hemoglobin molecule undergoes the polymerization under the low oxygen tension, causing the change in the shape of the RBC from by its biconcave disc to the elongated sickle-like structure. Next question. List the features of Drosophila melanogaster which makes it suitable for genetic experiments. So this Drosophila melanogaster that is used for the genetic experiments, what are the characters? Why this Drosophila melanogaster is selected? Because this they could be grown on simple synthetic medium in the laboratory. They can be grown in a simple synthetic medium. They complete their life cycle in about 2 weeks. So within a short period of time, they will complete their life cycle. So it is easy to do the experiments within a year. They can do the many experiments, many generation. So that is easy to grow the many generation within a month, within a year. And single mating could produce a large number of progeny flies. Okay, so the single mating, so the organisms they are produce the large number of progeny flies. Also, there was a clear differentiation between the sexes, that is males and female flies are easily distinguishable. So the males and females are they can be differentiated, and it has many types of hereditary variation that can be seen with low power microscope, the variation that can be seen in the low power microscopes. So these all are the important characters so that is present in the Drosophila melanogaster. So for that reason they selected the flies are selected for the experiment, genetic experiment. So one of the important question, ತುಂಬಾ ಸಲ ರಿಪೀಟೆಡ್ ಆಗಿ ಕ್ವಶನ್ ಅನ್ನ ಕೇಳಿದ್ದಾರೆ. ನಿಮ್ಗೆ 5 ಮಾರ್ಕ್ಸಿಗೂ ಇದನ್ನ ಕೇಳಬಹುದು, ಯಾಕಂದ್ರೆ 5 ಪಾಯಿಂಟ್ಸ್ ಬರೀರಿ ಅಂತ, 5 ಮಾರ್ಕ್ಸ್ಗೆ 5 ಪಾಯಿಂಟ್ಸ್ ಬರೀರಿ ಅಂತ ಈ ತರ ಕೇಳಬಹುದು. ಸೊ ಫಾರ್ 2 ಮಾರ್ಕ್ಸ್ ಯು ಕ್ಯಾನ್ ರೈಟ್ ಎನಿ 2 or 4 ಇಂಪಾರ್ಟೆಂಟ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್, ಇಂಪಾರ್ಟೆಂಟ್ 4 ಅನ್ನ ಬರಿಬಹುದು. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. ವಾಟ್ ಈಸ್ ಟೆಸ್ಟ್ ಕ್ರಾಸ್? Write its significance. A test cross is a cross between the unknown genotype and another homozygous recessive genotype. So test cross, when it is performed, for example, I have crossed with the tall plants with the dwarf plants. Okay. So the F1 generation, so what we get, that is all are TT, right? All are tall plants, but we have to see the gene. We don't know the genotype of these plants. Okay, so we don't know the genotypes of, are they homozygous or heterozygous? We don't know about that. To detect the plants, So unknown genotype. To detect the unknown genotype, it is crossed with the recessive. We know that this is the recessive plant, noun genetic, which is having the noun genetic combination. Then TT, we don't know is it dominant or is it homozygous or heterozygous. TT. Here, when we crossed this, we will get T, T, and T, 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 and this T, T, and T, T. Right. So here we got two uh, dwarf plants because it is a heterozygous. If it is a homozygous condition, if the unknown is a F1 generation is a homozygous condition, when we crossed with the recessive, all plants will be they are tall. But in the when we cross this unknown, we don't know it is homozygous condition or heterozygous condition. It is crossed with the um, recessive plant. So the two plants, they are tall, and two plants, they are dwarf. So these two plants, they when they grow, these two plants will grow as a dwarf. So we can conclude the F1 generation progeny that is having the heterozygous condition. Okay. What is the significance of this? Progenies of such cross can easily be analyzed to predict the genotype of the test organism. So we can detect the genotype of the test organism. Okay. So next question. What is meant by Mendel's law of independent assortment? So this is a law which is concluded by the Mendel's by performing the dihybrid cross. Here, the law states that when two pairs of traits are combined in a hybrid, segregation of the one pair of character is independent of the other pair of the characters. Here, when he crossed between the two characters, just take it as a tall or red flower forming plant and dwarf, dwarf white color flower, that is the recessive plant. So these two characters, so when these are the pair of characters when taken for the hybridization. So these pair of characters, so these characters, they will segregate each other with the independent of each other, because this T, so that can be combined with the another R. So like that, they are independent, the characters, they are independent of each other. Okay. So that is the law of segregation. They segregate, segregation of the one pair of the characters, that is independent, which is not depend on the other character which is present in that plant of the other pair of the characters. Okay. So this is the law of segregation. Next question. A man of blood group A marries a woman with blood group B. First child of a couple has O blood group. What other blood groups are expected to be in the future? Okay. So the man of blood group A. Blood group A means it can be IA I condition or IA IA condition. Alva. B blood group means they didn't mention what type of the, is it heterozygous or what? IB or IBI. Okay. Now, if I cross the homozygous, that is IA, just check it, IA and IB, there is a no chance of formation of the O blood group ಅಲ್ವಾ. ಇಲ್ಲಿ O blood group ಆಗ್ಲಿಕ್ಕೆ ಚಾನ್ಸೇ ಇಲ್ಲ, because all are AB blood group ಆಗತ್ತೆ ಅಲ್ವಾ. So I will take heterozygous A and homozygous B. Just check this condition. In this condition also, there is a no chance of the formation of the O blood group. O blood group that should have the I, alva, small I chain. But here there is IB, I will be there, B blood group will be there. So there is no chance of the blood group O. So I will take both are heterozygous condition, that is I crossed with, that is the B blood group woman, that is a heterozygous condition. Now check it once. IA, IB, IA, AB blood group. Then IA, small I, that is A blood group, IA, small I. Then small I, B, means IB, small I will write. Then last one, I. So, the the first child is having the O blood group. So the child is having the O blood group. There is a possibility of the having the O blood group. What the other groups are expected to be in the future? So in the future, if they have another child, there may be a possibility of the AB blood group, A blood group, and B blood group. Okay, there is a chance of or O blood group. So these are the expected blood group when they have the kid in the future. A, B and O blood group. Because both are heterozygous A and heterozygous B blood group. Next question. Why a cut results in non-stop bleeding in hemophilia? The possibility of female becoming hemophilic is extremely rare. Why? Here, the first question. In this disease, hemophilia disease, a single protein, that is a part of the cascade of proteins involved in the clotting of the blood, is affected. So, the single the protein cascade of proteins which are involved in the clotting of the blood is affected. Because of that reason, here the cut results in non-stop bleeding in the hemophilia. Then the possibility of the female becoming the hemophilic is extremely rare. Why? Because the possibility of a female becoming a hemophilic is extremely rare, because mother of such female has to be at least carrier, and uh, the father should be a hemophilic. Father hemophilic ಆಗಿತ್ತು, mother carrier ಆಗಿದ್ರೆ, there is a chance of the um, female to be a hemophilic. Okay. Write the causes and effects of phenylketonuria, PKU in shortly. What are the causes and effects of the phenylketonuria? So this PKU, phenylketonuria, this is inborn error of metabolism, is also inherited as the autosomal recessive trait. Here, the affected individuals lacks the enzyme that converts the amino acid phenylalanine into tyrosine. So that enzyme is affected because of this error, autosomal error. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. So here the phenylalanine, so that is accumulated and converted into phenylpyruvic acid. So here accumulation of these in brain result in a mental retardation, and these are also excreted through the urine because of its poor absorption by the kidney. So these all are the causes and effects. Why it is caused? Because of the accumulation of the phenylalanine and conversion of the phenylpyruvic acid, and when it is stored in the uh, when it is accumulated, so there is a chance of mental retardation. So that is the effects. Okay, one of the important question. So next we are discussing the 5-mark question. Okay, already we have completed the MCQs and 2-mark question. So that will be asked in your so that is the pattern of asking the question in your exam. So now very important 5 marks, because total 10 marks question will be asked from this session. Okay, here few autosome linked recessive blood disease occur in human population. Among them, some are related to the qualitative and quantitative problem of synthesizing the blood proteins. Explain. Here, autosome linked recessive disorder means, so it includes the sickle cell anemia, and thalassemia. Sickle cell anemia and thalassemia. Okay, so these are the autosome linked diseases which are related to the blood, where the qualitative and quantitative problem in the synthesizing the blood proteins. Here, one more, hemophilia, that is the sex-linked disease, which is not a autosome-linked disease. So here we have to explain about these two diseases. That is the 5-mark question. First, sickle cell anemia. How it is formed? This is a autosome-linked recessive trait that can be transmitted from parents to the offspring when both partners are carriers of the gene or heterozygous. So this disease is controlled by a single pair of genes, that is the HBA and HBS, that is the affected. Okay, that is the abnormal. Out of three possibilities of genotypes, only homozygous individual, that is if they are having the HBS and HBS condition, then the phenotype is affected. If they are having the heterozygous HBA and HBS, which is not affected. Okay, heterozygous individuals appear apparently unaffected, but they are the carrier. So they are the carriers, as there is a 50% probability of transmission of the mutant gene to the progeny, thus exhibiting the sickle cell trait. So they are transferring that from the parents to the next generation. So what happens if that gene is defected? The defect caused the substitution of the glutamic acid, so that is the amino acid. So the glutamic acid that is substituted by the valine, the change in the glutamic acid as a valine at the 6th position of the beta globin chain of the hemoglobin molecule. Okay, so there is a change in the glutamine. In place of the glutamine is changed. So the substitution of the amino acid in the globin protein results due to the single base substitution at the 6th codon. 6th codon ಏನಾಗಿರುತ್ತೆ beta globin chain ಅಲ್ಲಿ GAG ಏನಾಗಿರುತ್ತೆ GUG ಆಗಿರುತ್ತೆ ಅಲ್ವಾ. So the mutant hemoglobin molecule, so means which is having the which is shown a mutation alva, so that undergoes the polymerization under the low oxygen tension, causing the change in the shape of the RBC biconcave disc to the elongated sickle-like shape. Okay, so that is the sickle cell anemia. In short, you can write down the important points in this. Thalassemia. So here I will explain how it is caused, what are the effects. So this is a autosome-linked recessive blood disorder. Here it is transmitted from parents to the offspring when both partners are unaffected carriers for the gene, when they are in a heterozygous condition. The defect could be due to the either mutation or deletion, which is ultimately results in a reduced rate of synthesis of the one globin chain. So the quantitative, it may be alpha or beta chains that make the hemoglobin, that make up the hemoglobin. So there are two types of the thalassemia. The first type is the alpha thalassemia the alpha thalassemia production of the globin chain uh, alpha globin chain is affected, while beta thalassemia production of the beta globin chain is affected. Two types of the thalassemia. So here alpha thalassemia is controlled by closely related genes, that is HBA1 and HBA2 on the chromosome 16. So HBA1 and HBA2 chromosomes are affected of each of the parent, and it is observed due to the mutation or um, in the 16 chromosome ಮೇಲೆ ಏನಿದೆ HBA1 HBA2 ಅಂತ ಇದೆ ಅಲ್ವಾ, HBA1 HBA2 ಅಂತ ಇದೆ. The more genes are affected, less alpha globin molecules are produced. ಇಲ್ಲಿ ಜಾಸ್ತಿ genes affect ಆದ್ರೆ ಕಡಿಮೆ alpha globins production ಆಗತ್ತೆ. While beta thalassemia is controlled by single gene HBB on chromosome 11 of each parent and occurs due to the mutation of the both the genes. Okay. Then so you have to mention about the alpha thalassemia, beta thalassemia ಬಗ್ಗೆ explain ಮಾಡಬೇಕು. Thalassemia differs from sickle cell anemia in that former is the quantitative problem of the synthesizing too few globin molecules, while the latter is the qualitative problem of the synthesizing an incorrectly functioning globin. So both are related to the production of the protein in the blood. Okay, which are the blood related diseases. So here both you have to explain what is thalassemia, how it is caused, and what is alpha thalassemia and what is beta thalassemia. That is very important. Next question. Here, this is the model question paper question. Result of a Mendelian dihybrid cross are represented in the form of Punnett square. Here, the answer just observe this Punnett square and answer the following questions with respect to the results of the dihybrid cross. Okay, here just observe the Punnett square and tell me the answer. Number of parental types progeny. Number of parental types. Parental types means these are the parental type alva. F1 generation when it is cell pollinated, F2 is formed. So here number of progenies which are parental types. So let us check which are having the capital Y and small I, capital R and small r condition. So this one YYRR and 2, 3 and 4. So remaining, check it remaining. I think 4 is the correct answer. Number of parental types of progeny, that is 4. Wait. Yeah, 4. Alva, let us check the answer. That is 4. Parental types, that is 4. Okay. Then next question. Number of recombinant progeny. Here, the number of recombinants, which are the new recombinants are, how many recombinants are formed here? Total. So totally here, 8 recombinants are formed. 8 recombinant progenies are formed. Next question. Number of homozygous recessive progeny. Number of homozygous recessive. ಇಲ್ಲಿ ನೋಡಿ homozygous ಇದೆ, recessive ಇದೆ. So that is only 1. Homozygous recessive progeny, that is 1. Homozygous condition allide recessive ಇದೆ ಅಲ್ವಾ? So that is 1. Next question. Next question. Number of homozygous dominant progeny. Dominant ಇರಬೇಕು, homozygous condition, complete homozygous condition ಅಲ್ಲಿ ಇರಬೇಕು, ಚೆಕ್ ಮಾಡಿ. So this is the homozygous dominant. So remaining they all are the they are in a heterozygous condition alva. So idu homozygous recessive, this is the homozygous dominant. So only 1. I think 1 is the correct answer. Number of homozygous progeny for both the alleles. Okay, so there should be homozygous condition. So they should be they are in the homozygous condition for both the traits. Both the traits means here homozygous for the both traits. Just check it. So this is not a homozygous for ಇದು ಆರಿಗೆ ಹೋಮೋಸೈಗಸ್ ಇದೆ ಬಟ್ ಇದಕ್ಕೆ ಹೋಮೋಸೈಗಸ್ ಇಲ್ಲ ಅಲ್ವಾ ಇದು ಕೂಡ ಹಾಗೆ ಆಗಿದೆ ಇದು ಕೂಡ ಹಾಗೆ ಆಗಿದೆ ಸೊ ಆಲ್ ಆರ್ ಆಲ್ಮೋಸ್ಟ್ ಸೇಮ್ ಬಟ್ ಹೋಮೋಸೈಗಸ್ ಫಾರ್ ಬೋತ್ ದ ಟ್ರೇಟ್ ಇಲ್ಲಿ ನೋಡಿ ಇದು ವಾಯ್ಗೂ ಕೂಡ ಹೋಮೋಸೈಗಸ್ ಇದೆ ಆರಿಗೂ ಕೂಡ ಹೋಮೋಸೈಗಸ್ ಇದೆ ಒನ್ ನೆಕ್ಸ್ಟ್ ಚೆಕ್ ಮಾಡಿ ಸೊ ದಿಸ್ ಇಸ್ ಓನ್ಲಿ ಆರ್ ಇಸ್ ಹೋಮೋಸೈಗಸ್ ಇಲ್ಲಿ ಆರ್ ಮಾತ್ರ ಹೋಮೋಸೈಗಸ್ ಆಗಿದೆ ಅಲ್ವಾ ಇಲ್ಲಿ y r small r homozygous ಆಗಿದೆ ಫಾರ್ ಬೋತ್ ದ ಟ್ರೇಟ್ ಹೋಮೋಸೈಗಸ್ ಫಾರ್ ಬೋತ್ ದ ಟ್ರೇಟ್ಸ್ ಎರಡು ಕ್ಯಾರೆಕ್ಟರ್ಸ್ಗೆ ಬೋತ್ y ಮತ್ತೆ r ಗೆ ಹೋಮೋಸೈಗಸ್ ಆಗಿರೋದು ಓನ್ಲಿ 1 and 2. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ನಂಬರ್ ಆಫ್ ಹಿಟಿರೋಸೈಗಸ್ ಪ್ರೊಜೆನಿ ಫಾರ್ ಬೋತ್ ದ ಟ್ರೇಟ್ಸ್ ಎರಡೂದಕ್ಕೂ ಹಿಟಿರೋಸೈಗಸ್ ಆಗಿರ್ಬೇಕು ಸೊ ಹಿಯರ್ ಬೋತ್ ಆರ್ ನಾಟ್ ಹಿಟಿರೋಸೈಗಸ್ ಇದು ಕೂಡ ಒಂದಕ್ಕೆ ಹಿಟಿರೋಸೈಗಸ್ ಆಗಿದೆ ಹಿಯರ್ ದಿಸ್ ಈಸ್ ಹಿಟಿರೋಸೈಗಸ್ ಫಾರ್ ಬೋತ್ ಅಲ್ವಾ ಎರಡು ಕ್ಯಾರೆಕ್ಟರಿಗೂ ಹಿಟಿರೋಸೈಗಸ್ ಆಗಿದೆ ನೆಕ್ಸ್ಟ್ ಚೆಕ್ ಮಾಡಿ ಇದಾಗಿಲ್ಲ ಇದು ಕೂಡ ಆಗಿಲ್ಲ ಇದಾಗಿದೆ ಅಲ್ವಾ ಹಿಟಿರೋಸೈಗಸ್ ಫಾರ್ ದ ಬೋತ್ ಕ್ಯಾರೆಕ್ಟರ್ ನೆಕ್ಸ್ಟ್ ನೋಡಿ ಇದು ಆಗಿಲ್ಲ ಇದಾಗಿಲ್ಲ ಸೊ ದಿಸ್ ಒನ್ ಹಿಟಿರೋಸೈಗಸ್ ಫಾರ್ ದ ಬೋತ್ ದ ಕ್ಯಾರೆಕ್ಟರ್ ನಾಟ್ ದಿಸ್ ಒನ್. This is for the single character and this one and not this one. So total homozygous heterozygous for both the character. Y ಗೂ ಏನಾಗಿರ್ಬೇಕು ಹಿಟಿರೋಸೈಗಸ್ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಇರಬೇಕು R ಗೂ ಹಿಟಿರೋಸೈಗಸ್ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಇರಬೇಕು ಈ ತರ ಇರಬೇಕು ಅಲ್ವಾ ಸೊ 1 2 3 4 ಸೊ 4 ಆರ್ heterozygous for both the condition alva heterozygous progeny for both the traits. Number of pure line progeny is to pure line means which are not having the heterozygous condition ಇಲ್ದೇ ಇರುವಂತದ್ದನ್ನ ಪ್ಯೂರ್ ಲೈನ್ ಅಂತ ಕರೀತಾರೆ ಸೊ ದಿಸ್ ಇಸ್ ನಾಟ್ ಅ heterozygous and this one is not a heterozygous. Total 2 are not a heterozygous condition. They are called as pure line. Okay, next number of homozygous progeny for a single trait ಗೆ ಏನಾಗಿರ್ಬೇಕು homozygous ಆಗಿರ್ಬೇಕು. Okay, for single trait it should be homozygous. So this one for a single trait it should be homozygous condition. So this is also homozygous for the single trait and this is also homozygous for the single trait. Homozygous for the single trait. Here this is homozygous for both trait. I will not take this one. So this is homozygous for the single trait. And this one is also a homozygous for the single trait. This one. Okay. Here homozygous for the single trait ಏನಾಗಿರ್ಬೇಕು R R ಇರ್ಬೇಕು or R R ಇರ್ಬೇಕು Y Y ಇರ್ಬೇಕು ಅಥವಾ Y Y ಇರ್ಬೇಕು ಸಾರಿ small y ಇರಬೇಕು ಸೊ ಹಿಯರ್ ಅನದರ್ ಶುಡ್ ನಾಟ್ ಬಿ ಅ ಹೋಮೋಸೈಗಸ್ ಇನ್ನೊಂದೇನಾಗಿರ್ಬಾರ್ದು ಹೋಮೋಸೈಗಸ್ ಆಗಿರ್ಬಾರ್ದು ಹೋಮೋಸೈಗಸ್ ಫಾರ್ ದ ಸಿಂಗಲ್ ಟ್ರೇಟ್ ಸೊ ದಟ್ ಈಸ್ 1 2 3 4 5 6 7 8 ಅಲ್ವಾ homozygous for the single trait that is the 8. Sorry, homozygous, just a minute. Number of homozygous progeny for the single trait. So this one is completed. Number of heterozygous progeny for the single trait. ಸಿಂಗಲ್ ಟ್ರೇಟಿಗೆ ಏನಾಗಿರ್ಬೇಕು, ಹಿಟಿರೋಸೈಗಸ್ ಕಂಡೀಷನ್ ಇರಬೇಕು. ಸಿಂಗಲ್ ಟ್ರೇಟ್ ದಟ್ ಶುಡ್ ಬಿ ಹಿಟಿರೋಸೈಗಸ್. ನೋಡಿ, ಸಿಂಗಲ್ ಟ್ರೇಟ್ heterozygous condition ಅಲ್ಲಿ ಇರಬೇಕು. Single trait that should be a heterozygal condition. This is a single trait that is in a heterozygous condition, and this is also a single trait that is in a heterozygous condition, and this one is also single heterozygous condition. Single, that is a heterozygous condition. So like that you should find out the answer. Okay, so ಈ ತರ ನೀವು ಪನ್ನೆಟ್ ಸ್ಕ್ವೇರ್ ಅನ್ನ ನೋಡಿ ಆನ್ಸರ್ ಅನ್ನ ಫೈಂಡ್ ಔಟ್ ಮಾಡ್ತಾ ಹೋಗಿ. ನಂಬರ್ ಆಫ್ ರಿಸೆಸಿವ್ ಪ್ರೊಜೆನಿ ಫಾರ್ ದ ಸಿಂಗಲ್ ಟ್ರೇಟ್. ಸಿಂಗಲ್ ಟ್ರೇಟ್ಗೆ ರಿಸೆಸಿವ್ ಪ್ರೊಜೆನಿ ಎಷ್ಟಿದೆ, so that also you have to find out. So I hope you will check the last three answer. So check the checkerboard and write down the answer. Okay. Next question. Define the aneuploidy. Give two examples for this. Okay. Here, failure of the segregation of the chromatids during the cell division cycle result in a gain or loss of a chromosome is called a aneuploidy. Examples: Down syndrome. Down syndrome ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, gain of the one chromosome, that is the 21st chromosome trisomy ಆಗತ್ತೆ ಅಲ್ವಾ. And Turner syndrome, there is a loss of the X chromosome in human females, so that results total number of chromosomes 45. So these are the examples. And the second question nodi, the 3 mark question. Next, mention the karyotype of the Klinefelter syndrome and an affected individual and write its symptoms. So here, the genetic Klinefelter syndrome, that is the genetic disorder that is caused due to the additional copy of the chromosome resulting in the karyotype 47 ಎಷ್ಟಾಗತ್ತೆ because of the XXY ಅಲ್ವಾ. ಟೋಟಲ್ ಏನಾಗಿರತ್ತೆ, 44 pairs ಏನಾಗಿರತ್ತೆ ಸರಿ ಇರತ್ತೆ, ಬಟ್ ಡ್ಯೂ ಟು ದ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ XXY chromosome, so ಏನಾಗುತ್ತೆ total number of chromosomes that will be 47. So what are the characters? So they have the overall masculine development, however the feminine development also. So development of the breast and also expressed, so they are expressed as a sterile. So they are almost sterile. Next question. So thalassemia is a quantitative problem of synthesizing the globin. Explain in detail with respect to the chromosome number and genes and alleles and other features. So thalassemia ಬಗ್ಗೆ ನಾವು ಕ್ವಶನ್ ಅನ್ನ ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ದೇವೆ ಅಲ್ವಾ. So how it is caused, alpha type of the thalassemia, beta type of the thalassemia, and which are the chromosomes responsible for this 16th chromosome, and the alleles ಯಾವುದು, HBA, HBB ಅಲ್ವಾ. ಸೊ HBA1, HBA2, ಸೊ ಯಾವುದು alleles, ಸೊ ದಟ್ ಈಸ್ ಕಾಸಿಂಗ್ ದ ಡಿಸೀಸ್. ಸೊ ಇದನ್ನೆಲ್ಲ ನೀವು ಮೆನ್ಷನ್ ಮಾಡಿ ಈ ಒಂದು ಡಿಸೀಸ್ ಅನ್ನ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಬೇಕು. ಸೊ ದಟ್ ಕ್ಯಾರೀಸ್ ದ 5 marks. ನಾನು ಲಾಸ್ಟ್ ಕ್ವಶನ್ ಅಲ್ಲಿ ಅದನ್ನ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೇನೆ. ಪ್ಲೀಸ್ ಗೋ ಥ್ರೂ ದಟ್ ಕ್ವಶನ್. ಓಕೆ. Next question. Schematically represent the inheritance of flower in Snapdragon and draw the conclusion. So in the Snapdragon, so we know that incomplete dominance is occurred. Incomplete dominance, so that is represented as, so red flower, that is RR, and the white flower, that is taken as a recessive RR. They produce the gametes in the F1 generation. All flowers, they all are not red. If there is a dominance, they should produce the all red flowers, but pink color flowers means intermediate colors are formed because of the incomplete dominance. The dominant gene is not completely suppress the recessive gene. Because of that, pink color is formed. When these flowers are crossed, when these F1 generation is self pollinated, then They produce the F2 generation because they produce the R gametes and R gametes. So they produce the R R R R means these capital R small r, these all are the pink flowers, and this R is the red flower. So they produce no white flowers, they produce the one red flower. Sorry, they produce one, sorry, here R small r. Here two pink flowers and one white flower. Okay, so they are producing the one red flower, two pink flower, and white flower, that is also one. So that is the ratio you have to mention. ಫಿನೋಟೈಪಿಕ್ ರೇಷಿಯೋ ಮತ್ತೆ ಜಿನೋಟೈಪಿಕ್ ರೇಷಿಯೋನ ಮೆನ್ಷನ್ ಮಾಡಬೇಕು. ಚಕ್ಕರ್ ಬೋರ್ಡ್ ಅನ್ನ ಇಲ್ಲಿ ಹಾಕಬೇಕು. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. Give reasons for the following. A simple cut result in non-stop bleeding in the hemophilia in the affected individuals, because the reason for this, a hemophilia result in non-stop bleeding due to the lack of clotting factor. So that is the cascade protein is affected. The formation of the cascade protein is affected because of that. So that is the clotting factor. A simple cut result in a non-stop bleeding. Turner syndrome affected females are usually sterile. Turner syndrome causes the sterility due to the underdeveloped ovaries. Ovaries are not well developed because they are having the only one the chromosome number. So that is 45. That is because of the only sex chromosome X0. There is no XX chromosomes. Okay. So because they are having the underdeveloped ovaries. That is the second one. Third one. In Morgan dihybrid cross experiments on Drosophila showed that flies having the genes for yellow body and white eyes exhibited less recombination. Why less recombination occurred? Because they are due to the close proximity. They have the linkage. Close ಏನಾಗಿತ್ತು physically they are very near, so there is no recombination, there is less recombination occurred. Fourth one, inheritance of skin color in humans shows the different phenotypes, because skin color inheritance in humans shows the different phenotypes due to the polygenic inheritance. Last one, accumulation of phenylalanine in the body of phenylketonuria affected individuals. Here, phenylketonuria leads to the phenylalanine accumulation due to the deficiency in the enzyme which convert the that is the phenylalanine hydroxylase, which convert the phenylalanine into tyrosine. Okay. So these are the reasons, the 5 marks and 5 questions. So for 5 marks, 5 questions will be asked. Next question. Mention the chromosomal disorders that are due to the trisomy, represent their karyotype and two symptoms. So, trisomy, so that is occurred in the Down syndrome. That is having the 21st chromosome trisomy due to the 21st trisomy, and uh, Klinefelter syndrome, that is due to the trisomy of the sex chromosome, that is XXY chromosome. So the trisomy of these chromosomes. So what are the characteristics of these diseases? Very important. ನಿಮಗೆ ಈ ಡಿಸೀಸ್ ಬಗ್ಗೆ ಕೇಳ್ತಾರೆ. ಡೌನ್ ಸಿಂಡ್ರೋಮ್ ಡಿಸೀಸ್ ಸಿಂಪ್ಟಮ್ಸ್ ಮತ್ತೆ ಅದರ ಕ್ಯಾರಿಯೋಟೈಪ್ means you have to write how many chromosomes. The causes of this genetic disorder is that is because of the 21st chromosome trisomy. And here the affected individual is short, saturated with small round head, furrowed tongue and partially open mouth, palm is broad with characteristic palm crease, and physical, psychomotor, and mental development is retarded. So these all very importantly you have to write the characters. All characters you have to mention. Next one, Klinefelter syndrome. This genetic disorder is also caused due to the presence of additional copy of the X chromosome, that is forming the karyotype 47XXY. So here in this type of persons, they have the overall masculine development. However, feminine development is also there, and also these persons are sterile. These are the characteristics of the Klinefelter syndrome. Very important. Repeatedly, they will ask the question on this topic. Next question. What is aneuploidy? Differentiate between the aneuploidy and polyploidy. So aneuploidy andre gottide alva. So the failure of the segregation of the chromatids during the cell division, so that results the gain in or loss of the chromosome. That is called as the aneuploidy. And what is polyploidy? Polyploidy, um, so failure of the cytokinesis after the telophase stage. So the cell division result in the increase in the whole set of the chromosome in an organism. So this phenomenon is called as a polyploidy. Polyploidy commonly observed in the plants, and this aneuploidy, so that you give the example, uh, Down syndrome, Turner syndrome. So these are the examples. Describe the individual with the following chromosomal abnormalities. Here, ಇಲ್ಲಿ ನೋಡಿ trisomy of 21st chromosome, that is the Down syndrome ಅಲ್ವಾ. ಈ ಒಂದು ಡಿಸೀಸ್ ಬಗ್ಗೆ ನಾವು ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ, ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ ಅನ್ನ ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ. Down syndrome. Then XXY. XXY, that is a Klinefelter syndrome, that is also we have studied. Then X0, this is a Turner syndrome. ಇದ್ರ ಎಲ್ಲದ್ರ ಬಗ್ಗೆನೂ ನಿಮ್ಗೆ ಕ್ವಶನ್ ಅನ್ನ ಕೇಳ್ತಾರೆ. Turner's syndrome. Okay, so very important. Again, I will not explain this question. Already we have discussed this topic alva. Down syndrome, Klinefelter syndrome. This is about the Down syndrome, Klinefelter syndrome, and Turner syndrome ಅಲ್ವಾ. ಟರ್ನರ್ ಸಿಂಡ್ರೋಮ್ ಅಲ್ಲಿ ಏನಾಗಿರುತ್ತೆ, 45 ಕ್ರೋಮೋಸೋಮ್ಸ್ ಇರುತ್ತೆ ಬಿಕಾಸ್ ಆಫ್ X0, and females are sterile because of they have the rudimentary ovaries, and they also lack the secondary sexual characters. Next question. A colorblind father has a daughter with normal vision. Okay, a colorblind father is having the normal vision. The daughter marries a man with a normal vision. What is the probability of her children to be a colorblind? Nodi, a colorblind father. So I will take X, Y, has the daughter with normal vision. So daughter, I don't know, mother is carrier or what, I don't know. We don't know about that information. So but the daughter, she is having the normal um, normal vision. So she is not having the color blindness because mother is not a carrier. Mother carrier alla anta illi assume madona. Okay. The daughter marries a man with a normal vision. So here normal vision man. What is the probability of her children to be a color blind? See here, what is the probability of the, so this is the X, she's the carrier, and X, and man, he is having the normal vision. So there is a chance of X. Females are not affected, but there is a chance of affecting the sons. XX and XY. There is a explain with the help of the pedigree chart. Here, what is the probability of her children to be a color blind? So there is a 25%. There is a 25% of the probability of the her children will be a color blind, because if the son, he may or may not be have the color blindness because of this carrier mother. And the all one daughter may be carrier or not be a carrier. So this you have to explain in the pedigree chart. Idanna pedigree chart use ಮಾಡ್ಕೊಂಡು explain ಮಾಡಬೇಕು. Okay, so like this, male represented like this, female, and affected is represented in a black color. Next, there is a carrier, she is the carrier, and the normal man. Then what is the probability of the affected children, that you have to represent it in a pedigree chart. Nodi pedigree chart alli affected father ಈ ತರ fill ಇರೋದನ್ನ, and normal mother, and there is a chance of having the normal daughter marries a normal vision husband, but there is a chance of normal daughter, again normal affected son, normal carrier daughter, or normal son. So like this you have to explain. Okay, so idanna pedigree chart through ಈ ತರ explain ಮಾಡಬೇಕು. Next question ನೋಡಿ. A tall plant with red flowers. A tall plant with tall, I will take TT, with the red flowers, both are dominant character, are crossed with the dwarf plant. I will take TT, small t to represent the dwarf plant, and white flowers. So smaller. So you have to do the dihybrid cross. So in the F1 generation ಅಲ್ಲಿ ಏನಾಗತ್ತೆ, they produce the gametes TR and here TR. So these are the gametes. So you have to parental generation, gametes. F1 generation will be TT, capital R, small r. This is the F1 generation. When F1 generation is self-pollinated, F1 into F1, F1 generation is self-pollinated. So then the gametes production, so gametes they are produced is T, R, capital T, small t, capital R, and small t, small r. So these are the gametes produced. Using these gametes, you have to do the you have to find out the progenies in the F2 generation by using the checkerboard. Checkerboard ಅನ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಏನು ಮಾಡಬೇಕು, ಎಷ್ಟು ಪ್ರೊಜೆನಿಸ್ ಫಾರ್ಮೇಶನ್ ಆಗಿದೆ ಅನ್ನೋದನ್ನ, total number of type of the progenies form in the F2 generation, that you have to find out. ಇಲ್ಲೂ ಕೂಡ TR, capital T, small r, these are the gametes TR, TR, and TR, TR. Okay, so ಇದನ್ನ ಡೈಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಅನ್ನ ಚಕ್ಕರ್ ಬೋರ್ಡ್ ಥ್ರೂ ಎಕ್ಸ್ಪ್ರೆಸ್ ಮಾಡಬೇಕು. List out the characteristics of the chromosome theory of inheritance. Here, chromosomal theory of the inheritance. What are the characters? Here, the first is, during the meiosis, same chromosome pairs migrate as discrete structures independent of each other. That is the first characteristic. Chromosomes from each pair are randomly sorted. Each parent synthesizes gametes containing only half of their chromosomal material. Only a half chromosomal material will be there. Both male and female gametes contain an equal number of chromosomes. And fifth one, the produced offspring contain same number of chromosomes as their parents. The parental combination chromosomes are similar to the new progeny. Okay, so these are the characteristics of the chromosomal theory of the inheritance. Why is color blindness is more prominent in males than females? ಇದನ್ನ explain ಮಾಡಬೇಕು. So this is color blindness is sex-linked. It is sex-linked recessive disorder. Here, it is related to the X chromosome. In males, only X chromosome is there. So if the genes present in the X chromosome is defected, there is a chance of the male will be color blind. But the females, they are having the two X chromosomes. Because of that, if the genes present in one X chromosome is defected, another will be there. Two X chromosomes will be there. So because of that, males affect more prominently than the females. Okay, so that is the reason. Explain the law of dominance. So this law of dominance, so that you have to explain with example. You can give the example here. Uh, the characters are controlled by discrete units called factors, and factors are in pairs. In a dissimilar pair of character factors, one member of the pair is dominates, the other is the recessive. One is the dominant and other is the recessive. So this is the law of dominance. You can explain with the help of the example, monohybrid cross example. Okay, so ಇಲ್ಲಿ ನೋಡಿ same question ಇದೆ. Explain the gene inheritance with reference to the height of the characters. One gene inheritance, that is also a ಏನದು, ದಟ್ ಈಸ್ ಅ ಮೋನೋ ಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಅಲ್ವಾ. ಈ ಮೋನೋ ಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಥ್ರೂ ಏನ್ ಮಾಡಬಹುದು, ನೀವು ಲಾ ಆಫ್ ಡಾಮಿನೆನ್ಸ್ ಅನ್ನು ಕೂಡ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಬಹುದು. ಸೊ ದಿಸ್ ಈಸ್ ಅ ಒನ್ ಜೀನ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ದಟ್ ಈಸ್ ರಿಲೇಟೆಡ್ ಟು ದ ಟಾಲ್ನೆಸ್ ಆಫ್ ದ ಪ್ಲಾಂಟ್. ನೋಡಿ, ಹೋಮೋಸೈಗಸ್ ಟಾಲ್ ಮತ್ತೆ ಹೋಮೋಸೈಗಸ್ ಡ್ವಾರ್ಫ್ ಪ್ಲಾಂಟ್ ಅನ್ನ ತೆಗೆದುಕೊಂಡಾಗ F1 ಜನರೇಷನ್ ಆಲ್ ಪ್ಲಾಂಟ್ಸ್ ವಿಲ್ ಬಿ ಟಾಲ್. ಇನ್ ದ F2 ಜನರೇಷನ್ ವೆನ್ ದ F1 plants are selfed, so you will get a tall plant, three tall plants and one dwarf plant in the F2 generation. So you should write the phenotypic and genotypic ratio. Okay. So here, why I have taken this question, because you can explain the one gene inheritance and the law of dominance. So both using the monohybrid cross. Okay, monohybrid cross anna explain ಮಾಡಿದ್ರೆ law of dominance. So you can explain. So till now we have discussed the very important questions related to this chapter. Okay, I hope these questions that will be helpful for your board exam. Thank you. We'll meet in the next session.